Did you know that musical melodies, flashing lights and visual patterns, and even certain foods could trigger seizures in children diagnosed with Dravet syndrome? Dravet syndrome is a catastrophic form of pediatric epilepsy that causes intractable seizures in infants. Genetic studies have shown a strong association between the disorder and mutations of a gene called SCN1A. These mutations, which are not usually present in either parent, cause abnormalities in the function of brain cells, resulting in treatment-resistant seizures that occur in the first year of life of previously healthy babies. Although it is the fourth most common neurological disorder among infants, there is no known cure nor effective treatment for Dravet. Coupled with frequent seizures, children with Dravet suffer from intellectual disability and neurocognitive impairments that affect every aspect of their lives and place a devastating burden on their caregivers. Through a large-scale biochemical examination, scientists at Intellimedix are developing innovative algorithms to accelerate the discovery of new treatments for various neurological disorders, beginning with Dravet. By personalizing drug treatment to match individual genetic profiles, we aim to give the most beneficial preventative or therapeutic measures to each patient. Our drug discovery process is comprised of multiple steps. First, we determine the underlying genetic and molecular mechanisms of the disease in order to identify the genes that cause it. We do this at a personal level by sequencing the genomes of individual patients. Next, we develop computer models of these genes and structures to better understand the disease's method of action. We then perform a high-throughput in silico screening. In silico means performed on a computer or via computer simulation. We screen both FDA-approved and novel compounds against the mutated genes previously identified. The compounds that most successfully bind to their molecular target proceed to in vivo screening in animal models to determine physiological responses. At Intellimedix, we use zebrafish for modeling. These small fish, which originally come from the Himalayas, have become an important vertebrate model organism in genetic research over the past decade. Zebrafish are great models for drug discovery because their genome is 85% similar to the human genome. They are also physically transparent, easy to maintain, and susceptible to genetic modification and analysis. For our in vivo screening, zebrafish are created as general disease models or as a personalized model of a particular patient. When we build a personalized zebrafish model, we create a fish to have the same mutations as the patient in order to analyze the manifestation of the disease and identify optimal treatment for each individual. One of our main strategies throughout the screening process is drug repurposing, or finding new uses for previously FDA-approved compounds. Drug repurposing lowers both the risk and the cost of drug discovery, and more importantly, reduces the time it takes for the treatment to reach the patient. Of the 2.2 million people living with epilepsy in the U.S., as many as 15,000 have Dravet, and most do not respond to currently available anti-epileptic treatments. In fact, Dravet patients face a risk of sudden unexplained death from epilepsy that is 24 times higher than for patients with other types of epilepsy. Due to this heightened risk, debilitating seizures, and cognitive impairment, patients require constant supervision their whole life. Without a more effective treatment, the future for Dravet patients remains uncertain. Working with a team of world-class experts, Intellimedix is committed to developing innovative approaches to accelerate the discovery of drugs for Dravet and other neurological disorders, so we can bring new life to patients and their families.